Hello everyone, this is a D Pharmacy Pharmaceutics 1 class, subject code 1.1T, Unit 5. In this class, we are going to discuss rabies virus vaccines and typhus vaccines. So first of all, rabies vaccine. Rabies vaccines are white flocculent suspension in a clear liquid or slightly white to brownish white turbid liquid. Uh, it's a sterile suspension in saline or some other appropriate solution, which is isotonic with blood. Uh, Generally, here suitably killed rabies virus in uncontaminated brain tissues from animals uh, which are previously injected intracerebrally with the rabies virus. The preparation is It is prepared by injecting intracerebrally injecting to sheep, rabbit, sometimes suckling rats or mice or other animals with rabies virus. The animals which show paralysis, they are killed and their brains are harvested under aseptic conditions. Then it is tested for the absence of bacteria, and finally they are suspended in sodium chloride injection or any other suitable liquid. The suspension is inactivated by using phenol or beta propiolactone or any other means until the virus will no longer infect mice. It is diluted to produce a vaccine of the required strength. The vaccine is preserved by adding 0.01% weight by volume thiomersal, and the pH of the vaccine is adjusted between 7 to 7.2. Then it is transferred into sterilized containers and sealed. The vaccines must comply with the tests for sterility and the test for undue toxicity for vaccines. Storage of these vaccines, the vaccines should be stored in container protected from light and maintaining a storage temperature between 2 degrees centigrade and 8 degrees centigrade, and it should not be used until the absence of live virus has been established. Uses is used as prophylactic against rabies, uh, either for post-exposure immunization of person bitten by rabid animals or as a pre-exposure immunization of high-risk individuals. Dose of rabies vaccines are 1 to 5 ml daily for around 7 to 14 days according to the site and the severity of the bite and the risk of exposure to infection. Generally, it is given by subcutaneous injection. The next one we are going to discuss is typhus vaccine. We know that typhus vaccine is a sterile suspension of the killed rickettsia organism of a strain or strains of epidemic typhus rickettsia, which is known as rickettsia prowazekii. Rickettsia prowazekii is selected for antigenic efficiency. The preparation is initially it is prepared by injecting the virulent rickettsia into the yolk sacs of fertile eggs which have been incubated for 7 days. Within a period of 9 to 13 days after injection, the yolk sacs are collected under aseptic conditions, and then it is subjected to suitable treatment for liberating the maximum number of rickettsia. The material is suspended in normal saline solution, and formaldehyde solution is added into that. The concentration is between around 0.2 to 0.5%. The suspension containing 10 to 15% weight by weight of yolk sac tissue is purified with solvent ether, and the aqueous middle layer is collected and it is distributed into sterile containers under aseptic conditions. The vaccine must comply with the test for sterility and the test of undue toxicity for vaccines and sera. The vaccine can also be prepared from the lungs of small rodents uh, in which rickettsial pneumonias have been caused by inhalation of large doses of virulent rickettsia or by giving intraperitoneal injections of rickettsia. Storage, it should be stored between 2 to 8 degrees centigrade, and uses, as you know, that it is used as a prophylactic against the epidemic typhus, and the dose also is a prophylactic agent to prophylactic by subcutaneous injection, and the dose is specifically 0.25 to 1 ml. So that's it for this class. uh, in my next class I will be discussing different toxoids like diphtheria toxoids and other toxoids. So thank you.